In the tapestry of human knowledge, few threads are as profound, as pervasive, and yes, as tangled as the story of life itself. For millennia, this story remained a mystery, a cosmic secret whispered only in the rustling of leaves, the blossoming of flowers, and the birth of new life. But within the past century, we have deciphered this code, mapping the very essence of what makes us us, exploring the grand symphony of life, encoded in our DNA. We begin our tale not in a laboratory, but in the timeless expanse of human contemplation. Long before the dawn of science, long before the language of genetics filled our textbooks, there existed a profound human impulse, the seed of curiosity. For them, the legacy of their ancestors flowed through bloodlines, carried on the backs of eagles and snakes. Here amidst the olive groves and sun-drenched marble, some of the greatest minds in history pondered the nature of life and its Hippocrates, the father of medicine, ventured a hypothesis that challenged the prevailing notions of his time. Ayurveda and Taoist philosophies viewed heredity through the lens of balance and flow. Still, throughout the medieval period, the question of inheritance continued to intrigue thinkers. Though their inquiries were often filtered through the lens of religious belief, the balance of dashas in Ayurveda and the harmonious interplay of yin and yang in Taoism both emphasized the importance of inherent qualities passed down from previous generations. His tools were simple. Pea plants, an array of traits such as color, shape, and size, and an unwavering dedication to observation and data. Through meticulous breeding experiments involving thousands of pea plants, Mendel uncovered fundamental principles of inheritance. The question that had first taken root in ancient contemplation had been whittled down to a single: what molecule carries genetic information? He introduced concepts like dominant and recessive traits, laying the groundwork for what we today call genetics. And stay tuned for the rest of our series as we continue to explore the fascinating world of genetics and the incredible discoveries that have shaped our understanding of life itself. As the 1900s dawned, the hunt for the substance of life intensified. Though groundbreaking, their discovery went largely unnoticed. The protein establishment was too entrenched to be dislodged. The leading contenders were proteins, complex molecules celebrated for their structural roles in the body. But could they also carry the blueprints of life? Its discovery in 1957 by Arthur Kornberg marked a significant leap. The true potential of DNA lay dormant, waiting for the right conditions to unfurl. Enter Marshall Nirenberg. In 1961, he cracked the genetic code, deciphering how DNA's four-letter sequence translated into the language of life. DNA polymerase provided the key to understanding how genetic information was copied during cell division, a process vital to inheritance. At the heart of this transformation are two pivotal figures, James Watson and Francis Crick. In 1953, armed with the X-ray diffraction images of DNA provided by Rosalind Franklin, together they paint a picture of life's blueprint, a testament to the power of scientific inquiry. And with the understanding of DNA's structure and function, a new set of questions arises, setting the stage for the next chapter in our journey. If you found this content insightful, don't forget to subscribe to stay tuned for more captivating explorations into the world of genetics. And Maurice Wilkins, these scientists unveiled the double helix structure of DNA, a revelation that shook the foundations of biology. This discovery earned them the Nobel Prize in 1962. Or was that role reserved for a simpler, seemingly insignificant molecule? Despite his groundbreaking work, Mendel's findings remained largely unrecognized during his lifetime. If you found this content valuable, please consider subscribing. Your support helps us continue to create educational content that brings the wonders of science to curious minds like yours. Until next time, keep asking questions, keep exploring, and remember, the answers you seek may be closer than you think, but the seeds of his discoveries had been sown, and they would soon bear remarkable fruit. The seeds of curiosity, long dormant, are beginning to sprout, nurtured by a growing interest in the natural world and the burgeoning field of biology. The stage is set for a scientific revolution. The microscope became a window into the inner workings of the cell, revealing mysterious thread-like structures within the nucleus, later named chromosomes. Scientists began to piece together the puzzle of heredity, recognizing that chromosomes played a crucial role in transmitting genetic information. By the turn of the 20th century, the stage was set for a monumental breakthrough. With the rediscovery of Mendel's work, genetics as a field of study was born, and as we move towards the mid-1800s, we find ourselves in the company of an unassuming monk-slat, Gregor Mendel, who would forever alter our understanding of genetics. While not scientific by modern standards, these systems were nonetheless remarkable for their keen observation and logical framework. He proposed that the various parts of the human body, from the humors that coursed through veins to the tissues that formed the very structure of the organism, all contributed to the creation of offspring. This was a revolutionary idea, a departure from the prevailing belief that placed sole emphasis on the role of the male in procreation. According to Hippocrates, the female provided merely a passive vessel, a notion that would persist for centuries. These mythical narratives, while rooted in religion, were not entirely disconnected from observation. He believed that the male reproductive organ, with its ability to move and penetrate, harbored the essence of life, a miniature homunculus that would develop into a complete human being. While scientifically inaccurate, this idea profoundly impacted the Western understanding of genetics, shaping debates that would rage for centuries. They recognized patterns in nature, resemblances between parents and children that hinted at a more tangible reality beneath the layers of myth. It was a simple, fundamental wonder that echoed across civilizations and through the ages. How do children resemble their parents? This was not a casual query, but the genesis of a much larger question, a question that would eventually lead to the very core of life itself. So join me as we embark on a breathtaking journey through 14 astonishing chapters, each marking a pivotal moment in our understanding of genetics, a testament to human curiosity and scientific brilliance. This is more than just a history, it's a celebration of the relentless pursuit of knowledge that drives us forward, together. From the microscopic world of genes to the ethical frontiers of genetic engineering, each chapter is a new revelation, a step closer to unraveling the secrets held within ourselves, and who knows, maybe along the way, we'll discover just how extraordinary life itself truly is. Welcome to Chapter 1 of our 14-part journey into the scientific triumph that redefined what it means to understand life itself. As we delve into the annals of history, we find this primordial question taking root in the fertile ground of human imagination. In ancient Mesopotamia, where the cradle of civilization nurtured the first stirrings of abstract thought, the concept of heredity was often intertwined with the realm of the divine. To the Mesopotamians, the transmission of traits from parent to child was seen as a reflection of the will of the gods, a testament to the divine essence imbued within each individual. Their myths wove tales of gods, mingling their own attributes into the fabric of offspring, shaping their destinies with each passing generation. Across the vast expanse of the ancient world, in the heart of Mesoamerica, the Aztecs developed their own rich mythology to explain the mysteries of inheritance. However, it was in ancient Greece that we find perhaps the earliest serious attempt to grapple with the riddle of heredity, but it was Aristotle, the polymath, who shaped the course of Western thought, who would present the most influential idea yet. In stark contrast to the prevailing Greek philosophy, ancient Indian and Chinese traditions offered a different perspective. By the Middle Ages, however, science took a back seat to theology, and the seeds of curiosity began to wither under the weight of dogma. Fast forward to the 19th century, and the intellectual climate begins to shift once again. Mendel's experiments in the monastery garden were not conducted in the shadow of towering labs or with the aid of cutting-edge technology. Then came the late 19th century, a period of rapid advancement in biological and chemical research. And so we arrive at the brink of the 20th century with the basic principles of heredity established, but without a clear understanding of the molecule responsible for carrying genetic information. This brings us to the beginning of Chapter 2. Join me as we dive deeper into the scientific quest to uncover the substance of life. And thank you for joining me on this enlightening journey through the history of genetics. In the quiet solitude of the laboratory, under the glow of Bunsen burners and microscopes, a revolution was brewing. In 1944, a landmark experiment conducted by Oswald Avery, Colin MacLeod, and Macklin McCarty pointed to deoxyribonucleic acid, better known as DNA, as the likely candidate. However, as we step into the 1950s, the stage is set for a transformation. But even as the significance of this breakthrough resonated, another crucial player emerged, the enigmatic enzyme DNA. Yet despite these momentous discoveries, the story doesn't end here. These discoveries, spanning from the structure of DNA to its function and replication, form the bedrock of molecular biology. As we conclude this chapter, remember, every great scientific breakthrough sparks countless questions. And with that, we come to the close of our exploration of Chapter 3. This has been a journey through the intricate mechanisms that drive the essence of life. Until next time, keep questioning, keep exploring, and remember, the answers you seek may lie within the very essence of life itself. 1 billion tiny bells are ringing within each of your cells right now, triggering reactions that make life possible. They're called enzymes, proteins that accelerate the chemical processes that keep you alive. You can thank them for digesting your food, replicating your DNA, fighting off infections, and until the mid-20th century, they were the leading suspects in solving the riddle of heredity. Proteins seemed to fit all the clues. They were made of amino acids, and their long, complicated structures suggested a molecule with limitless potential, ideal for carrying genetic information. But proteins turned out to be red herrings, leading scientists up one blind alley after another. While enzymes themselves were still something of a mystery, their importance was clear. Scientists in the late 19th and early 20th centuries were just starting to understand that every function in a cell seems to depend on some kind of enzyme. Enzymes drive metabolism, the full set of chemical processes that occur in living cells. And those processes start with taking in raw materials, like sunlight or nutrients, and releasing energy in forms that living things can use. This was just one area where biology was making great strides. Others included physiology, the study of how organisms function, and histology, the study of tools like the electron microscope were opening up new worlds within cells, allowing scientists to see not just their outlines, but their internal structures. As they peered deeper into the workings of cells, the old idea of the cell as a simple sack of chemicals began to seem more and more inadequate. Cells, it turned out, were complex chemical factories humming with activity. New fields like biochemistry and cell biology were born, and with them, the question, how do cells work? At the center of that question was the mystery of heredity. Scientists were beginning to understand that genetic traits are transmitted through discrete units called genes. but they knew almost nothing about how genes carried information. There was also a major rift in the scientific community about whether genes were physical things or abstract qualities. Some argued that genes were particles, while others insisted that they were simply properties of cells. For example, the gene might be nothing more than a region of the cell that has somehow become specialized. A small difference, perhaps, but one with profound implications. Was the hereditary material a physical thing that could be studied scientifically, passed around like a ball during a game of catch? Or was it some kind of force, like gravity, that pulled certain traits out of cells? If it was the latter, then there would be no way to pin it down. So in many ways, the biggest breakthrough of the era was the rediscovery in the early 1900s of Gregor Mendel's laws of inheritance. Working with pea plants in the mid-1800s, the Augustinian monk had uncovered patterns in how traits like color and shape are transmitted from parents to offspring. But the real significance of his work was not appreciated until 3 scientists independently rediscovered it. In 1900, suddenly biologists had a framework to begin testing the physical basis of heredity. Was the genetic material a particle or a property? To answer that, they needed to find it. And to do that, they needed more tools. So they turned to unlikely allies, borrowing equipment and ideas from other fields, and built entirely new ways of looking at cells and molecules. Enter the physicist. Not everyone was comfortable with this intermingling of disciplines. Some biologists were suspicious of physicists, seeing them as armchair theorists who preferred grand pronouncements to careful observation. But these criticisms did little to dampen the enthusiasm of brilliant minds like Niels Bohr, who in 1933 suggested that life might have its own physical principles. 3 years later, his student, the Danish physicist Bohr, wrote his PhD thesis on the conservation of symmetries in biological processes. And in 1944, the legendary physicist Erwin Schrödinger gave a series of lectures at Dublin University titled, "Yes, What is Life?" The stage was set for a revolution. The years between the world wars saw massive advances in technology and intellectual exploration. Major scientific discoveries like penicillin and the structure of the atom captured the public imagination, but they also led to new kinds of destructive technologies, from radar to the atomic bomb, prompting people to wonder, what else don't we know? Perhaps most important, these were years of intense intellectual cross. Scientists no longer stuck strictly to their specialties. Instead, they shared ideas across disciplines, creating explosive new combinations of thought and research methods. This trend was especially evident in biology, as physicists like Bohr and Schrödinger brought their fresh perspectives. Biologists found themselves asking fundamental questions about how life works. How does a single fertilized egg turn into a complex organism? How do cells replicate? How does genetic information travel between generations? What is life? By the late 1940s, these questions were attracting some of the brightest minds in the world. Armed with powerful new tools, they were ready to crack one of nature's most important codes. By the 1930s, a new generation of instruments had made studying molecules more precise and accessible than ever before. Early instruments, like the ultracentrifuge, separated cell components based on their size and density, yielding pure samples of DNA, RNA, and proteins. Spectrophotometers allowed scientists to analyze the amount and types of light molecules absorbed, giving clues about their structure. But it was X-ray diffraction that really advanced the field. By the late 1930s, scientists like William Astbury and his students were producing the first detailed pictures of molecular structures. This technique involved firing X-rays at crystallized samples and analyzing the resulting diffraction patterns. which revealed information about the molecule's atomic arrangement. This gave them an unprecedented window into the molecular world. Though it was a bit like looking through a foggy window pane. These images were blurry and hard to interpret, but scientists were nonetheless excited by what they saw, particularly the helical shapes emerging from the data. Astbury, for instance, suggested that proteins might twist into regular spirals, a finding that would later inform our understanding of alpha helices, a common structural motif in proteins. Though at the time, no one suspected that the greatest application of X-ray crystallography would come from a different kind of helix altogether. Meanwhile, in California, Linus Pauling was using entirely different tools to produce stunning results. Pauling's work centered on chemical bonds, the glue that holds molecules together. His classic textbook, General Chemistry, introduced concepts like electronegativity, which measures an atom's ability to attract electrons and form bonds. Armed with these ideas, Pauling developed some of the earliest models of molecular structure. But his interests soon turned to more controversial topics. Specifically, the nature of the chemical bond in proteins and DNA. Proteins, Pauling argued, must be held together by more than the simple bonds described by traditional chemistry. He hypothesized that interactions like hydrogen bonding play a crucial role in their structure. But even as he made waves in the scientific community, he missed one critical detail: the exact structure of DNA. That revelation would come from an unlikely source. Bacteriophages infect bacteria by injecting their genetic material through the cell wall. Because they are so simple, consisting of just a protein shell and some genetic material, they became an ideal model organism, or other model virus, for studying biology at the molecular level. By the 1930s, a group of scientists known as the Phage Group were investigating them in a dedicated laboratory at the California Institute of Technology, led by Max Delbrück, Salvador Luria, and Alfred Hershey. These researchers included future Nobel laureates and some names you may know today, like Seymour Benzer and Francis Crick. Together they explored how genes replicate and mutate, laying the groundwork for molecular biology. In fact, the Phage Group's work with these tiny machines helped shift attention away from the mysterious inner workings of cells and toward the molecule previously thought too boring to matter: deoxyribonucleic acid. Now that scientists had the tools to isolate and study DNA, interest in its structure was skyrocketing. But here was the problem. No one knew what it looked like. Proteins were still the front-runner in the search for the hereditary material, and DNA was still considered too simple a molecule to carry genetic information. But one scientist was skeptical. Having read Schrödinger's book, What is Life? As a teenager, James Watson became obsessed with solving the structure of DNA. In 1951, Watson left his job as a biologist at the University of Indiana to join Francis Crick at Cambridge University's Cavendish Laboratory. There they began their investigation using every tool available, including X-ray diffraction images taken by Rosalind Franklin. Meanwhile, across the hall, another team, led by Linus Pauling, was closing in on the answer. The race was on, and it was just getting started. The quest to understand DNA was attracting top talent from around the world, and at the heart of this scientific gold rush were two institutions: Cambridge University's Cavendish Laboratory and the California Institute of Technology. Their proximity to each other and to Birkbeck College in London, where Franklin worked, meant that new findings could quickly spread and inspire competition. And it was in this atmosphere of friendly rivalry that ideas like the double helix began to take shape. The winner of the race would receive immense prestige, but there was much more at stake. Unraveling the structure of DNA was not just about winning a prize or publishing a paper. It was about changing the world. It promised insights into everything from heredity to evolution, to the origins of life itself. The stakes had never been higher. In the quiet moments, when inspiration struck, in the heated debates during late-night talks, and in the meticulous work of analysis, these brilliant minds were pushing the boundaries of human knowledge. Each step brought them closer to the blueprint of life, and with it, they were poised to unlock secrets that had been hidden for billions of years. And thank you for watching. Chapter 3. London's Lab Wars. Franklin, Wilkins, and the Shadow of Discovery. It's misunderstood. If Cambridge was a cauldron of ideas and ambition, then King's College London was a crucible of tension. It was a battleground of personalities. John Randall, the department head, had appointed both Franklin and Wilkins to work on DNA, but with vague and overlapping responsibilities. Franklin, unaware that Wilkins considered himself the lead researcher, assumed she had full independence. Wilkins, meanwhile, saw her as an assistant forced on him. His early images of DNA hinted at helical symmetry, though they were far less refined than he received the Nobel Prize for discovering the structure of DNA, but he never won widespread recognition as a lone investigator. Shared. On January 1953, James Watson visited King's College. It contained the molecule's dimensions about 20 angstroms in diameter, and with bases stacked 3.4 angstroms apart. The tragedy here wasn't theft, it was miscommunication, institutional hierarchy, and a culture that undervalued dissent, especially from women. As the next chapter unfolds, we head to Cambridge and watch Watson and Crick begin their model building, race-shuffling cardboard bases, arguing over symmetry, and inching closer to a structure that would change the world. But ultimately, it was also a case of being in the right place at the right time. Just like Maurice Wilkins, we operate in the shadows, providing critical support for important scientific work. Our supporters help us keep our videos free for everyone. And in return, we've got some great perks to say thanks. So whether you're a student, educator, or just a fan of cool visuals, we've got something for you. And as always, thank you for watching. Franklin wasn't hidden from the discoveries. She was central to it, but the timing of her conclusions and the networks around her meant that others published first. Watson and Crick would use the data to their advantage and eventually win the Nobel Prize. Franklin, meanwhile, would soon leave King's for Birkbeck College, where she'd make major discoveries about viruses before her death in 1958. Wilkins would remain at King's, sharing the 1962 Nobel Prize with Watson and Crick. These dimensions would prove invaluable for Watson and Crick. Without Wilkins's photograph and Franklin's measurements, the race for the double helix might have continued for years. But now it was down to a sprint. The meeting was arranged by John Randall, who thought the young American might benefit from seeing Wilkins' sDNA images. Instead, Watson saw something that made his eyes pop. Maurice Wilkins, feeling a bit awkward about showing the photos without Franklin's permission, quickly added, "Of course, Miss Franklin would have liked to show you these photographs herself." But Watson barely registered those words. The image confirmed what he and Crick had suspected but had been able to visualize: a helical structure with precise symmetry and repeating patterns that matched a double-stranded spiral. Wilkins, ever conscientious, asked, "Do you think it's a spiral?" Watson replied, "It looks like one to me." But Watson didn't just see an image. He saw a model waiting to be built. It gave him the missing link he needed to complete the double helix, yet his contributions were vital. Wilkins was among the first to apply X-ray diffraction to DNA in biological form, a technique Franklin further refined. He recognized the importance of helicosymmetry, though he didn't pursue it as aggressively as Franklin. He believed in collaboration but was trapped in hierarchical assumptions that prevented him from fully embracing it. When the double helix was proposed, he supported Franklin, but he didn't insist that she be included in the accolades. He was, after all, a product of his time. His actions, though sometimes criticized, were often driven by a desire to advance science and mentor young researchers. He admired Franklin's work but struggled to communicate with her. This wasn't because he didn't recognize her brilliance, but because of his own limitations. Plus, the rigid academic culture of the time left little room for anything but top-down direction. So while Wilkins appreciated Franklin's skills, he couldn't deviate from his boss's orders, which included keeping a close eye on her work. And that brings us to Photo 51. In 1953, feeling isolated and unsure, he shared Photo 51 without Franklin's knowledge or permission with James Watson during a visit to King S. This wasn't a casual slip. Wilkins believed he was acting on behalf of Randall, who wanted feedback from the young upstart at Cambridge. But it would be an understatement to say that Franklin was furious. She considered the move a betrayal and a disservice to science. But like many of her complaints, they fell on deaf ears. The situation was further complicated by Randall, who favored Wilkins and failed to mediate between them. It was here, in the dim corridors and basement laboratories of the biophysics department, that some of the most important experimental work on DNA was unfolding, not with dramatic eureka moments, but with painstaking precision. The air was filled with cigarette smoke and unspoken rivalries, and at the center of it all were two people destined for a fractured partnership: Rosalind Franklin and Maurice Wilkins. Let's start by setting the stage. This is the story of Photo 51 and the strange tale of its legacy. And if you're new here, welcome. We've made this series to explore the stories behind some of the most iconic discoveries in science. Okay, let's get into it. 3.1 A-Lab. Divided, the King's College Biophysics Unit in the early 1950s wasn't a place of shared vision. Franklin was sharp, principled and uninterested in small talk. She brought with her a mastery of X-ray crystallography and a tireless commitment to experimental rigor. Their personalities clashed immediately. Not out of malice, but misunderstanding. Wilkins was quiet, introverted, and cerebral. He had worked on radar during the war and now turned his attention to nucleic acids. With a chemistry PhD from Cambridge and pioneering work on carbon and graphite behind her, she arrived at King's in 1951, ready to dive into biological mysteries. She brought with her an X-ray camera of superior design. By the standards of the day, her lab was cramped and poorly equipped. But Franklin didn't grumble. She got to work. She even went so far as to install a device to eliminate tremors from nearby streets, a level of thoroughness that was almost unheard of. Her colleagues noted her near-obsessive standards, her unwillingness to accept sloppy results or rushed conclusions. She was meticulous about eliminating every possible source of error or contamination in her experiments. She prepared her own samples with exacting care, and she even built a special rotating anode to improve the quality of her X-ray images. What other researchers needed days to collect, Franklin often managed in hours, and her DNA images were sharp, elegant, and deeply mysterious. They revealed something extraordinary about the molecule's structure, a hint of helicosymmetry, but they were also frustratingly ambiguous. Franklin wasn't just taking pictures. She was developing mathematical interpretations of the diffraction patterns. Deciphering them was as much an art as a science. She pursued multiple possibilities, sometimes contradicting herself. She even deduced, ahead of anyone, that DNA likely had two helical forms, depending on humidity, aDNA and bDNA. And in May 1952, she captured Photo 51, a crystalline, ghost-like image of B-form DNA that whispered a secret no one had yet understood. 3.2 Rosalind Franklin. The invisible force? To call Rosalind Franklin brilliant is an understatement. 3.3 Maurice Wilkins. The quiet pioneer. Maurice Wilkins, despite his challenges working with Franklin, was no slouch. In reality, Maurice Wilkins was a pioneer, but he always operated in someone else's shadow. First Randell's and then Franklin's 3.4, the missing link. Around the same time, another key piece of Franklin's work, a report she had written, was shared by Max Parrots at the Medical Research Council. 3.5 A Collision of Worlds. The story of Photo 51 had begun. So had the swirl of debates over credit, sexism and scientific ethics that would follow for decades. We'll see you next time. Thanks for watching. This episode was brought to you by our supporters. Hey, this is someone from my channel. You may know me from Minute Earth. And yes, we're still working on the whole audio thing. I am so excited to be bringing you guys a science podcast. We will meet brilliant minds and learn how their personal stories intersect with one of the most significant scientific endeavors ever. Let's get started. In our series, we'll explore what really happened in the race to discover the structure of DNA, the molecule of life. Discovery is a collision of curiosity, obsession, and just the right amount of arrogance. But after being inspired by Erwin Schrödinger's What is Life, his gaze shifted from feathers to genes. He earned a PhD. Despite skepticism from more senior scientists, they quickly realized they complemented each other. Watson brought instinct and clarity. Crick brought structure and strategy. They were outsiders to the field, and perhaps because of this, they were willing to challenge assumptions. Watson and Crick spent the next year reading, speculating, and refining ideas behind the scenes. Okay, that's actually not true. Now we have to build suspense. Are they gonna succeed? Will the model hold up? How will Linus Pauling react? Stay tuned to find out. They began studying the chemical bonds of nucleotide bases and contemplating base-pairing rules. Our team has been hard at work on this series for months now, and we are so excited to finally be able to share it with you. This project was truly a labor of love. We wanted to tell the story of the discovery of DNA in a way that was dramatic, engaging, and educational. So we hired actors to bring the characters to life, composed an original score, mixed in sound effects, and more. They read Pauling's papers, anticipating the moves of the only scientists they feared. You can also write a review on whatever platform you're using to listen to this. Reviews help other people discover new podcasts. Also, if there's someone who you think would love this story, please send them this episode. Sharing is caring. Finally, consider supporting us. We're a small team and your support means a lot. Thanks for listening. And when 1953 arrived with new clues in hand from Photo 51 and Franklin's unpublished data, they were ready to act. The final model was near. The spiral was beginning to unwind. Most other scientists tried to solve DNA through experimental data. Watson and Crick did something radical. They tried to build it as a physical model. Model building, once dismissed as child's play, was about to become the key to unlocking life. He had studied physics, worked on mine warfare during World War II, and transitioned to biology, believing that great mysteries lingered in the tiny world of molecules. It looked elegant, but was chemically impossible. Phosphates are negatively charged. Clumping them together at the center created repulsive forces. They had placed bases on the outside, ignoring the hydrophobic nature of these structures. When Rosalind Franklin visited Cambridge and saw the model, she shredded it verbally. She pointed out its basic flaws and criticized their lack of understanding of X-ray It was a public embarrassment, They were told by Sir Lawrence Bragg, head of the Cavendish, to back off the DNA problem and let the King's College team handle it. And they did. For a while, Crick was loud, persuasive, and brimming with theories. He loved talking, often to the exasperation of colleagues. He was fascinated by molecular symmetry and information theory, concepts that would later help conceptualize DNA's structure as a carrier of information. Though he hadn't yet finished his doctoral work, Crick had a mind that moved faster than credentials, and when he met Watson, the spark was immediate. Dee, at just 22, working on bacteria, and soon sought to uncover the structure of DNA, an ambition that led him across the Atlantic to the Cavendish Laboratory at Cambridge. He wasn't a chemist, nor a crystallographer, but he was bold and unafraid of making mistakes. His intuition was his compass, and he believed you didn't need to know everything, just the right things. Arriving at Cavendish, he was instantly drawn to one man, Crick. In 1951, two very different men crossed paths in Cambridge. One was a lanky, enthusiastic 23-year-old from the American Midwest with a rapid-fire voice and infectious ambition. The other was a loquacious 35-year-old English physicist who hadn't yet completed his PhD. Together, they would challenge the giants of biology and race toward the molecule at the center of life. They were James Dewey Watson and Francis Harry Compton, Crick, names that would become inseparable from the double helix. 4.1 James Watson. The American outsider James Watson had grown up in Chicago, more interested in birdwatching than test tubes. 4.2 Francis Crick. The theorist without a thesis, Francis Crick was twice Watson's age, British and intellectually relentless. 4.3 A partnership born of obsession. Watson and Crick clicked over conversations about helical symmetry, base pairing, an X-ray diffraction often, held over lunch, or shouted across the Cavendish lab. 4.4 Building the first models. In 1951, the duo constructed their first model of DNA, a triple helix with phosphate groups at the core. 4.5 Waiting in the wings, but the obsession didn't fade. And now you know what happened next. But wait, before you go, I have some news to share. So how can you help us share this incredible story with even more people? Well, first, make sure you subscribe. In 1952, two boffins in Cambridge got wind of an image taken by a brilliant scientist at King's College London. This image, which resembled nothing so much as a child's drawing of a spiral staircase, would soon become the blueprint for our understanding of life itself. Over the course of just a few weeks, these two scientists would use the image to construct a model of one of nature's most important molecules, a molecule which carries the genetic instructions for all known living things. They would discover the secret of life. Well, maybe not quite, but they would certainly discover the secret of DNA. This is the story of how it happened. On the other end is a photographic film, which will record the results. The X-rays pass through the crystal and some are scattered, a phenomena known as diffraction. But none of them were successful because the technology available at the time was insufficient. This began to change with the advent of newer, more powerful X-ray sources and detectors. Imagine a long, thin thread and wrap it around a spool. One of the difficulties was that DNA comes in two different crystalline forms, A and B. Form A is shorter and wider than B, and scientists didn't know what caused the difference. But Franklin was more precise. She used a mathematical technique known as a Fourier transform to determine the positions of atoms in a crystal. Although the image clearly showed a helical structure, she didn't want to jump to conclusions. To be fair, it's unclear whether anyone else did. At the time, although scientists suspected that DNA was helical, they couldn't be certain. So photo 51 remained something of an enigma until one day when Franklin's colleague, Raymond Gosling, pointed out that the crossed arcs in the image suggested that there were 2 repeating units per turn of the helix. But she should be remembered as the woman who cracked the code of life. This detail alone was enough to convince some scientists, like American physicist Charles Wilkins, that the helix was correct. By using this approach, she was able to obtain highly detailed images of DNA. She continued to push for more data and more patterns before coming to any conclusions. Ironically, she would never get the chance. Other accounts say that Wilkins stole the image from Franklin's lab and showed it to Watson. Regardless, what Watson saw when he looked at the image is what really matters. It showed the X-shaped pattern he and Crick had long suspected was true. All of a sudden, the structure of DNA snapped into place. The rest, as they say, is history. Photo 51 never quite left the lab at King's College, but copies of it made their way to the Cavendish Laboratory at Cambridge, where James Watson and Francis Crick were trying to work out the structure of DNA. Her best-known image was called Photo 51, taken in May 1952. It showed the X-shaped pattern characteristic of a helical structure and provided strong evidence that DNA had a helical structure. The impact of their discovery is impossible to overstate. Thanks to them, we now know how genetic traits are passed from parents to offspring, how mutations occur, and how genetic engineering works. What's more, this was problematic, because if you wanted to build a model of DNA, you needed to figure out which form it took. By 1950, scientists were aware that the form existed, and they had managed to get a picture of it, but getting a good image was extremely difficult. Then imagine the thread split into 2. Each half now has the ability to act as a complete thread. This is the essence of DNA's structure, which is why it's called a double helix. This makes DNA particularly useful as a storage system. In fact, if you wanted to store as much information as the human genome on a single floppy disk, you would need a data density of approximately 1 terabyte per square inch. And that's just crazy. But more importantly, it changed when one scientist at King's College London named Rosalind Franklin took over the college's new X-ray department and started working on DNA. The scattered X-rays produce spots on the film, and each spot represents a different orientation of a crystal lattice. This was due to her unique methods. First, she built a better X-ray camera capable of producing higher quality images. Second, she developed techniques to keep the DNA samples hydrated, which allowed her to observe both forms, A and B, and third, she brought mathematical rigor to the analysis. When all the orientations have been accounted for, you have a full diffraction pattern. What's more, the intensity of each spot tells you how many electrons were diffracted at that angle. Since electrons are bound to atoms, this gives you information about the positions of the atoms in the crystal. If you want to understand the very essence of life, then you need to understand DNA. This is pretty straightforward if the crystal is big enough, but the smaller the crystal, the harder it is to analyze. With small molecules, like those of salts or sugars, for example, you can simply analyze the diffraction pattern to work out the structure directly. There she developed expertise in the technique was well suited to Franklin. Like many scientists, she enjoyed the methodical precision of the lab. According to her biographer, she liked to do everything herself. Collect and analyze the data, run the numbers, draw the graphs. In general, Franklin was a stickler for detail, and her work ethic was relentless. This proved to be crucial for cracking the structure of DNA. But with bigger molecules, like DNA, it's not so easy. DNA is a particularly challenging molecule to analyze because it's both big and amorphous, meaning its atoms lack an obvious repeating pattern. This means that Even if you manage to get a good diffraction pattern, i.e., a clear image, it's still really hard to decipher the structure. We don't have time to get into all the boring details here. But basically, DNA contains our genes and is the thing that makes us us. For most of history, however, we didn't know what DNA even was. It wasn't until 1953 that James Watson and Francis Crick uncovered the structure of DNA, the twisting ladder depicted in school textbooks today. Even before that, though, we had known for some time that DNA was a kind of molecule called a polymer, made up of smaller units, joined together like beads on a string. We also knew that these units, called nucleotides, came in for varieties, denoted by the letters A, T, C, and G. We didn't know, however, what the structure of DNA was or how these four nucleotides combined. The first step towards finding out was to analyze its structure using a technique called X-ray. The process begins by purifying a sample of DNA and then placing it inside a device called a centrifuge. The centrifuge spins the tube at high speed, forcing the DNA to settle at the bottom in a crystalline form. This is already tricky business, since the quality of the image you get depends entirely on the quality of your crystal. DNA is a long and flexible molecule, however, so getting it to settle into a stable crystal is easier said than done. If the DNA is too dry, it tends to tangle. If it's too wet, it takes on a different shape. As you'll see later, this is a bit of a problem. Once a suitable crystal is obtained, it's placed at one end of an X-ray beam. From this information, you can work out the structure of the molecule by trial and error, making a physical model until it fits. In the late 1940s and early 1950s, a number of groups around the world attempted to use X-ray crystallography to study DNA. Franklin was born in 1920 in New York and studied chemistry at Cambridge University before moving to Paris to work under the famous chemist Jacques. But before we get to that, let's take a step back and explain what Franklin was working with: DNA. Although we now know how amazing DNA is, at the time, scientists were still struggling to get a handle on it. And this is where Franklin's expertise lay. Whereas other scientists had struggled to produce even a vague image of DNA, Franklin produced a number of crisp, well-defined ones. Until Franklin's day, scientists had relied on educated guesses to interpret the patterns they observed in the films. Now, it's important to point out that Franklin wasn't so sure about this herself. It seems that Franklin, however, was not among them. There are conflicting accounts of how this happened. According to Watson, he stumbled upon the image while visiting Wilkins, who was showing it to him to boast. Watson and Crick went on to build a model of DNA, publish their findings, and win a Nobel Prize. As for Franklin, she died of cancer in 1958 at the age of 37. She never won a Nobel Prize, nor did she receive much public recognition during her lifetime. Did you enjoy this video? Then check out our video on how DNA works. Brilliant is the best way to learn math, data science, and computer science interactively. They offer thousands of lessons across 65 courses, from foundational and advanced math to AI and neural networks. Whether you want to know how to make informed decisions in business or understand the code behind computer graphics, Brilliant has you covered. Their interactive courses help you learn complicated concepts by letting you play around with them. You'll get a chance to apply what you learn in real time and solidify your understanding of even the most challenging topics. Learning something new can feel overwhelming, but with Brilliant, you can go at your own pace, learn on the go, and even earn badges for the skills you master. I've been learning a lot about large language models on Brilliant lately. These are the neural networks behind tools like ChatGPT and Google Bard. I learned how these models use deep learning to generate realistic responses to natural language inputs. After taking this course, I'm a lot more informed on one of the hottest topics in tech today. Thanks to Brilliant for supporting this video. Chapter 6. A game of models. Piecing together the spiral to solve the unsolvable. Sometimes you just have to build it. February 1953. The lab at Cavendish hummed with quiet tension. Across a workbench, atoms made of wood and wire hung from rods like skeletal dreams. Watson began constructing nucleotide bases: adenine, thymine, cytosine, and guanine, each designed to rotate and bond. Crick kept hammering out geometric and symmetry rules, ensuring that whatever they built would conform to X-ray data and chemical logic. Every day brought tweaks. Every night brought new speculation. One problem remained. How did the bases pair? Why didn't they just bond with nearby? Two scientists, neither of them crystallographers, neither trained in biology, once again breaking the rules of scientific decorum. His work on DNA, while overshadowed by the race to discover its structure, was crucial in providing key pieces of information about its helical nature. Though his contribution is often overlooked, Donohue's knowledge of chemical bonding and molecular structure was instrumental in solving the final piece of the puzzle. These were giants whose shoulders Watson and Crick stood on. Their work and brilliance paved the way for one of the most significant scientific discoveries of the century. Without Wilkins' contribution of Photo 51 and the dimensions of the helical rise, Watson and Crick might have struggled to assemble all the pieces of the puzzle. Rosalind Franklin was a brilliant scientist whose skill in X-ray diffraction produced the highest quality images of DNA. While her data was critical to Watson and Crick, she was largely excluded from the small circle making decisions about its interpretation. Sadly, Franklin passed away in 1958, unaware of the impact her work would have on the world. That information reshaped Watson's thinking. He built cardboard cutouts of the correct molecular structures and began playing with complementary hydrogen bonding. Every piece fit. They had solved the structural riddle of DNA. In the next chapter, we'll see how Watson and Crick presented their findings to the world. If you're re-enjoying this series and want to support us in reaching more people with our science storytelling, please consider becoming a patron. Your support will help us grow our team and continue producing high-quality educational content. Thank you for watching. See you next time. But that solution would change the world only if they could prove it. 6.5 Shouting in the Eagle. On February 28, 1953, Crick reportedly burst into the Eagle, a Cambridge pub, and shouted, "We have found the secret of life." It was premature, perhaps, and yet not wrong. They had built a working model that fit the data and explained genetic replication. What began with paper and intuition had crystallized into a structure that would redefine biology. Then it clicked. A paired with T through 2 hydrogen bonds. G paired with C through 3 hydrogen bonds. Perfect fits, identical shape, and crucially, symmetry preserved within the helical framework. Watson rushed to Crick. "It's got to be like this." Together, they constructed a full 3-dimensional double helix with complementary base pairs hidden inside. Phosphate backbones on the outside, spiraling upward like a twisted ladder. It was beautiful, and it made sense, but this time they were getting it right. The game of models was back on, and the stakes were everything. In Part 3, we saw Watson and Crick score critical data from Rosalind Franklin and Maurice Wilkins. But first, let us take a moment to appreciate the contributions of those whose work paved the way for this breakthrough. Linus Pauling was a towering figure in chemistry. His work on chemical bonds revolutionized our understanding of molecular structure. But his interests weren't confined to the lab. He was an outspoken advocate for peace and a critic of nuclear proliferation, earning him the Nobel Peace Prize in 1962, though he would later come to regret it. Pauling initially dismissed DNA as a structural chemist, focusing instead on proteins. Yet his 1951 book, The Nature of the Chemical Bond, provided the foundation for understanding the forces that hold biomolecules together. Now they need to translate that data into the first accurate model of DNA's structure. 6.1 back in the ring following their failed triple helix attempt in 1951. James Watson and Francis Crick had been instructed to keep away from DNA, but that unofficial exile ended in early 1953 when Rosalind Franklin's data, including Photo 51, came into their view. With Wilkins' unintentional leak and Max Parrott's shared MRC report, their workspace wasn't a crystalline lab, it was a chaos of paper clips, rulers, rods, and wooden balls. 6.3 The base pairing revelation. The breakthrough came from a quiet visit to Jerry Donoghue, a visiting chemist at Cambridge, who corrected Watson's mistaken assumptions about base tautomers. How T, G, and C molecules naturally align. They tested it all. X-ray diffraction, chemical compatibility. In Chapter 7, the Eureka moment, we'll sit beside them in the Cavendish office as they prepare their no-legendary paper and race to publish before Linus Pauling does. Maurice Wilkins was a physicist turned biophysicist, instrumental in bringing X-ray crystallography to bear on biological problems. Jerry Donoghue was a chemist who played a pivotal role in nudging Watson towards the correct base pairing in DNA. And as we move forward to the next chapter, I hope you'll join me in appreciating the countless others whose names we may never know, whose work and brilliance also shaped this monumental discovery. Let's test your understanding. Question: what does the structure proposed by Watson and Crick have in common with a staircase? Well, it's a twisted ladder. How did they come up with this idea? They looked at existing evidence and imagined something that fit. That sounds reasonable, but is it scientific? Well, scientists are people too, so they can get things wrong. In 1968, a book titled The Double Helix, a personal view of the discovery of the structure of DNA, was published. Franklin was a pioneer in the field of X-ray crystallography, and her work was instrumental in providing the key pieces of evidence that led to the discovery of the double helix. Often it comes down to differences in personality, like the fact that Franklin was reserved while Watson and Crick were eager for attention. Sometimes it's unconscious bias, meaning that people don't even realize they are favoring someone else because of stereotypes about gender roles. For example, then there's bad behavior. Watson and Crick weren't shy about criticizing Franklin's work, even though some of her unpublished data was crucial to their discovery. And sometimes it's simply bad luck. Franklin died of cancer in 1958, before the full impact of the discovery was widely recognized. Franklin's X-ray diffraction images, especially Photo 51, provided the first visual evidence for the helical structure of DNA. Credit and recognition are not always given equally. That's unfortunate. History tends to favor those who are assertive and visible, but it doesn't mean that everyone's contributions are equally valuable. In fact, sometimes the opposite is true. Rosalind Franklin never won a Nobel Prize, but her contribution to the discovery of the double helix is undeniable. In recent years, there has been a growing movement to recognize and celebrate the contributions of women in science. Franklin is one of several women whose stories are being rediscovered and celebrated. The discovery of the double helix was a monumental achievement that changed our understanding of life itself. But it was also a human story with all of the complexities and imperfections that come with it. Watson and Crick's story gets most of the attention. But it is important to remember that they were not the only ones involved, and that their contributions were shaped by the work of others, including the often overlooked Rosalind Franklin. Without her work, Watson and Crick would not have been able to develop their model. Despite her critical contributions, Franklin's name was largely absent from the scientific papers describing the double helix, and she was excluded from many of the public celebrations that followed. This was partly due to the sexism of the time, and partly because Watson and Crick were more comfortable with the limelight. However, in recent years, Franklin's contributions have been more widely recognized and celebrated. Several books and articles have been written about her life and work, and her image is featured on the periodic table of elements. The book painted a picture of the discovery process as a competition between two teams, one at Cambridge led by Watson and Crick, and the other at King's College London led by Franklin and Wilkins. And Watson portrayed himself and Crick as the heroes of the story, downplaying the contributions of others, particularly Franklin. The double helix caused a storm of protest from scientists who took issue with Watson's characterization of Franklin as a pedautor, and his failure to acknowledge her significant contributions. One scientist who worked closely with Franklin called the book a distortion of history of the worst kind. And Franklin's close friend and colleague, Anne Barrington, said that Watson's account was malicious gossip presented as science. However, despite the controversy surrounding the book, it caught the public imagination and sparked interest in the discovery of the double helix. It remains one of the best-known accounts of the discovery, has been credited with helping to launch the careers of several of the scientists involved. But when they review each other's work, they also use reason and evidence. So what counts as evidence? In science, ideas need to be You can't prove an idea correct, but you can show that it fits with the evidence you have, or that it predicts outcomes that are later confirmed. In the case of the double helix, the model was built using existing measurements and images like Photo 51. And it predicted that DNA should look a certain way under an electron microscope. These predictions were soon confirmed. So the double helix stuck around. The publication of the double helix changed the course of biology and ushered in the era of molecular biology. For Watson and Crick, it was also the start of fame. They accepted invitations to speak all over the world and were even interviewed by the press. But it was far from a smooth road to recognition. The Nobel Committee. Watson and Crick were nominated for the Nobel Prize in Physiology or Medicine in 1956. You know, that prize awarded annually to scientists who have made the most significant discoveries in physiology or medicine, but they didn't win. Maybe the committee felt that the significance of the discovery hadn't yet been fully realized. Or perhaps they thought more time was needed before awarding such a prestigious prize. Two more nominations followed in 1958 and 1960, but still no Nobel Prize. Then in 1962, they won, along with Maurice Wilkins. They shared the Nobel Prize in Physiology or Medicine for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. The award was well deserved, but there was one notable absence: Rosalind Franklin. While the press release announcing the award mentioned all three contributors, the actual Nobel Prize medal went only to Watson, Crick, and Wilkins. And if that snub wasn't enough, Franklin's family did not even receive a letter informing them of the award until 3 weeks after the ceremony. Public perception, despite the Nobel Committee's recognition. Many years passed before Watson and Crick received widespread public acclaim. Franklin's contribution, while Watson and Crick rightfully received much of the credit for discovering the double helix. They would have been the first to acknowledge the crucial role that Rosalind Franklin played in the discovery. Now let's think about why stories like Franklin's happen. There are multiple explanations. So here's the moral of the story. I hope you found this helpful. Sometimes winning means more than medals, and losing means more than obscurity. In the months following their publication in Nature, James Watson and Francis Crick were vaulted into scientific stardom. The double helix was no longer a model, it was a revelation. Soon, biology departments, pharmaceutical labs, and universities were swarming with researchers, diving into DNA like prospectors in a molecular gold rush. But as the light of fame intensified, shadows stretched longer, and one of the darkest fell upon the legacy of Rosalind Franklin. It caused an immediate stir. He portrayed himself as impulsive and bold, Crick as brilliant, Wilkins as reluctant, and he depicted Franklin dismissively referred to as rosy, as prickly, humorless, and slow to see the truth. Many saw this as deeply unfair. Colleagues who knew Franklin spoke out in defense of her brilliance, kindness, and professionalism. You can find these exercises in the exercises section of this chapter. These interactive exercises will help you to absorb all the information you've learned in this chapter. Remember, learning is not a passive process. We appreciate your feedback, and we're always looking for ways to improve our videos. Also, remember to subscribe so you won't miss any of our latest updates. You must engage with the material to truly understand it. So go ahead and give these exercises a try. It sparked a movement to reclaim Franklin's place in the story, not as a helper, but as a pioneer. We have many more videos of the DNA Learning Center on this platform. Our mission is to provide free science education for everyone. We believe that everyone has the potential to understand and appreciate science. With your support, we can continue to create and share our science videos and other educational resources. So please consider making a tax-deductible donation. Together, we can make a difference. Thank you for watching. The accolades begin by 1954. The Watson-Crick model was being cited around the world. The DNA boom had begun. They worked with data passed to them without consent, but in a scientific culture less conscious of intellectual property. Should Franklin have shared the Nobel? Possibly. But her early death complicated the question. Is recognition just about prizes? Or is it about how history is told and whose voices are amplified? Over time, Franklin's reputation grew not through awards, but through legacy. Institutions renamed buildings, films and plays celebrated her story, and female scientists worldwide drew inspiration from her perseverance in a field stacked against her. Textbooks were updated overnight with new diagrams of the double helix. Experimentalists rushed to test the implications of base pairing and replication. Molecular biology was no longer a fringe field. It was now the future. Francis Crick, ever the theorist, gave lectures tying their model to genetic coding. James Watson traveled across the US, riding a wave of recognition. Their once dismissed model was now being hailed as the most important biological discovery of the century. The Nobel Prize. The pinnacle came in 1962, almost a decade after the 1953 papers. The Nobel Prize in Physiology or Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins, the citation, for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. Rosalind Franklin was not among the recipients. She had died 4 years earlier, in 1958, from ovarian cancer at age 37. The Nobel Prize does not grant awards posthumously, but even in life, she hadn't received the acclaim her peers now enjoyed. What did she know? Franklin wasn't unaware of Watson and Crick's model. In fact, by the time it was published, she had independently come to very similar conclusions about the helical structure, but had not yet finalized or published them. At Birkbeck College, where she had moved after King's, She made foundational discoveries in virology, including crystallographic studies of the tobacco mosaic virus and poliovirus. She was building a second legacy when illness took her. Still, for years, she remained a footnote in the DNA story. It would take books, feminist scholarship, and historical reassessment to change that. The Double Helix, a book and a reckoning in 1968, James Watson published The Double Helix, a personal and unfiltered account of the discovery ethics of recognition. Historians and scientists have since grappled with the moral complexity of DNA's discovery. Did Watson and Crick cheat? In Chapter 9, Cracking the Genetic Code, we will explore what came next, how the double helix became a Rosetta Stone for biology, and how the language of life was deciphered, letter by letter. Now let's do some exercises to check your understanding. And if you want to learn more about amazing scientists, don't forget to subscribe. Below are some suggested videos that you might enjoy. Don't forget to click on the like button and write a comment before you go. Chapter 10, Rosalind Remembered. Shadows, science, and a shifting spotlight. She was not a muse, a footnote, or an assistant. She was a scientist, brilliant, exacting, and essential. For decades, the name Rosalind Franklin lingered at the edges of the DNA story, whispered in footnotes, glimpsed in acknowledgments, but never given full voice. Yet over time, a movement grew, not just to celebrate her legacy, but to reframe the entire narrative. This wasn't revisionism, it was revelation. This chapter is her chapter. The scientists behind the lens, Franklin was never seeking stardom. What she wanted, what she demanded, was clarity, accuracy, and intellectual integrity. Those who worked with her, from technicians to peers, felt the weight of her standards. But they also recognized her fairness. If you made a good solution or solved a tricky problem, she would praise you unreservedly. It was just that the bar was high, and sometimes people weren't ready to reach it. This applied doubly so when those people were senior male colleagues. At the time, the academic world was a tough terrain for women, especially Jewish women. There was an expectation that they would publish prolifically early in their careers, then scale back as they got married and had children. For Franklin, this wasn't an option. She wed twice, but childbearing wasn't part of the equation. By all accounts, she was brilliant but abrasive, demanding and disagreeable. And while these traits made her an excellent scientist, they made her a difficult colleague. As historian of science Margaret Rossiter put it, she was not a nice person. No one would have been surprised if she had ended up working alone. That she didn't was a testament to the dedication and curiosity of the people she worked with. And nobody was more curious than James Watson. Though their relationship was often contentious, he too was undeniably influenced by Franklin's exacting nature. When he saw photo 51, he says he could immediately tell it was hers. He knew that no one else could have produced such clear, meticulous work. It was a quality that set her apart, but also left her vulnerable. By the time Watson and Crick proposed the double helix, the spotlight was firmly fixed on them. Franklin's contribution was relegated to an acknowledgement, but as time went on, the full force of her intellect could no longer be ignored. Her work in X-ray diffraction was among the most advanced of the era. While others in the field focused on protein structures, she was obsessed with nucleic acids. Not only did she meticulously study both A DNA and B DNA, determined to isolate their features, she also helped prove the helical nature of the molecule. And contrary to popular belief, she was among the first to suggest that DNA's phosphate backbone lay on the outside, not the inside of the helix. This insight, based on analysis of photo 22, was crucial to Watson and Crick's eventual model, because it meant the negatively charged phosphates could interact with positively charged proteins, helping package the enormous molecule into a compact nucleus. They didn't know it yet, but without her work, their discovery wouldn't have been possible, and it wasn't as if Franklin was unaware of what they were doing. The three of them had lunch together on occasion, and Watson frequently visited her office. Yet the idea that she might have played a key role in the discovery didn't take hold until much later. According to historian of science John Emsley, Photo 51 was not accidental genius. It was the result of hours of calibration, patience, and mathematical interpretation. Franklin produced some of the clearest X-ray diffraction images of DNA, but getting them required painstaking work, something Watson and Crick didn't fully appreciate. After King's College in 1953, Franklin left King's College London for a new position at Birkbeck College, London, where she joined J.D. Bernal's crystallography lab. Bernal was a prominent advocate for using science to address social issues and had recently published work arguing that the structure of matter was key to understanding life itself. Franklin spent her time here applying her expertise to viruses, which she felt were on the borderline between living and non-living. She became the first to reveal the RNA structure of the tobacco mosaic virus and uncovered its helicar symmetry. Although the technique was similar to that used to study DNA, few people at the time appreciated the connection. Franklin was uncovering foundational knowledge for structural virology that would influence modern research on viruses like HIV and influenza. It may have taken decades to catch on, but her contemporaries did recognize her groundbreaking work. Despite limited resources and frequent sexism in the field, she published prolifically and was elected a Fellow of the Royal Society in 1957, the year before her death. And she mentored other researchers, among them Mr. Aaron Klug, who would later win a Nobel Prize himself for work on the structure of nucleosomes, the complex of DNA and proteins that makes up chromosomes. When Franklin died, he was just starting out in his career, and her loss profoundly affected him. "I never really got over it," he said. "It was a terrible shock. Illness and silence." In 1956, Franklin was diagnosed with ovarian cancer, possibly linked to early lab exposure to X-ray radiation before adequate protections were standard. Even after surgery, she continued to work, taking long sick leaves but still publishing papers based on data collected during her treatments. In 1957, she was given a clean bill of health, but the following year, the cancer returned. She underwent further surgery and chemotherapy, which rendered her partially blind. Now, facing the reality of her situation, she took on a new determination to secure her place in the field. She traveled to the United States in late 1957 to give a series of lectures and meet with colleagues she had only corresponded with before. By all accounts, the trip was a success. She made valuable connections, won new friends, and came home feeling buoyed and optimistic about the future. Back in London, she reconnected with her friends, Francis Crick and his wife Odile, who played a key role in supporting her through her final months, arranging for transportation to the hospital and helping her keep up with her correspondence. In April 1958, at the age of just 37, Franklin died. By then, the double helix had been accepted as the basis of the genetic code, and the race to understand it was in full swing. But she never knew how central her legacy would one day become. The posthumous rise, like many historical narratives, the story of DNA tends to center around the accomplishments of white men. But as posthumous scholarship has shown, it was shaped by contributions from across the globe. As recently as the 1990s, scientists were recognizing Franklin's critical role in elucidating the structure of the helix. In 1975, American author Anne Sayre published the first biography of Franklin's life. The book, Rosalind Franklin and DNA, rebutted Watson's portrayal of her as a cold and distant colleague, and instead painted a portrait of a warm, funny, and self-aware woman who was passionate about science. Then in the 1990s and 2000s, there was a wave of renewed academic and public interest in her life and work. Since then, buildings, research centers, and scholarships have been named after her, and she has become a feminist icon. In 2015, Nicole Kidman portrayed Franklin in Photograph 51, a West End play by British playwright Anna Diva Smith that brought her story to the stage. Finally, after decades of footnotes and blurred images, she had become front and center. More than recognition today, Rosalind Franklin stands as a scientific icon. Her name adorns the Rosalind Franklin Institute, a world-leading center for interdisciplinary research in the life sciences, as well as the European Space Agency's next Mars rover slated for exploration missions in the 2030s. Long after her own discoveries, her brilliance continues to illuminate the way forward. Beyond her scientific excellence, Franklin represents the cost of invisibility, what it does to those who are erased from history and to the history they help create. Her story serves as a reminder that we must constantly interrogate whose voices are missing and ensure that their contributions get their rightful place in the narrative. As Franklin herself wrote, science and everyday life cannot and should not be separated. Her life embodied that belief. Her lens revealed the helix, but her story reveals us. Ready for Chapter 11? In the next part, we'll explore the science unleashed by the helix to birth of biotechnology, PCR, and the quiet revolution unfolding in petri dishes and startups alike. In 1953, DNA was a structure. By the 1970s, it became a tool, and by the 21st century, it was a revolutionary shaping medicine, agriculture, computing, and even ethics. Molecular biology goes mainstream. In the wake of Watson and Crick's discovery, labs worldwide shifted their focus to understanding DNA's role in replication and expression. They raced to crack the genetic code, those 4 letters spelling out life's instructions. By the 1960s, powerful tools emerged to study this code. In 1973, Paul Burke inserted a monkey virus gene into E. coli bacteria, creating one of the earliest genetically modified organisms. Its name itself was a fusion of genetic engineering and technology, signaling a shift in how science and commerce intertwined. Genentech was co-founded by Boyer and venture capitalist Robert Swanson, who recognized the vast potential of DNA, inspired a wave of startups blending science with Silicon Valley's risk-taking ethos. By the 1980s, biotech was a booming industry. What began in academic labs now sat in stock markets, courtrooms and clinics, and it wasn't just about money. People felt the ethical earthquake caused by biotechnology. For diagnostics, PCR enabled the detection of minute traces of viruses or pathogens, leading to earlier and more accurate disease identification. Even in fields like evolutionary biology, where ancient DNA is the subject of study, PCR proved invaluable. In Chapter 12, Ethics, Genes, and Power, we will dive deep into the ethical dilemmas and profound questions raised by these technologies. What does it mean for a scientist to play God? What are the limits of genetic modification? And finally, here are your suggested readings. If you want to dive deeper into the topics discussed in this chapter, you can also explore additional resources to expand your understanding of the molecular age. Remember, you can find all these materials and more in the provided resources. Thanks for joining us in this chapter. If you enjoyed this content, please don't forget to subscribe for more. Your support helps us continue exploring the fascinating world of science and technology. And how do we ensure the responsible use of these powerful tools? Join us always as we navigate the complex landscape of ethics, genes, and power in the age of biotechnology. It allowed researchers to work with degraded or minute samples, opening up new avenues in understanding ancient life forms. This chapter explores how DNA went from being a scientific curiosity to a powerful tool and eventually a revolution in biotechnology. We saw how scientists went from discovering the structure of DNA to manipulating it and replicating it, ushering in the age of genetic engineering. We also looked at some of the key milestones during this period, including the development of recombinant DNA technology, the birth of the biotech industry, and the invention of PCR. Additionally, we discussed the broader impact of these technologies, including their commercialization and the ethical questions they raised. PCR didn't just change how experiments were conducted. It changed what was possible. It gave scientists the ability to work with minuscule samples, opening up new frontiers in genetics. Their timing couldn't have been better. By the mid-1970s, the US government had relaxed regulations on biotechnology, creating a fertile ground for startups. This decision unleashed a firestorm. The idea that someone could own a life form created in a lab challenged fundamental views about nature, ownership, and even life itself. Genentech went public in 1980, raising over $80 million in the process, much of it based on the potential of DNA technology. Kary Mullis, a biochemist at Cetus Corporation, invented the polymerase chain reaction, or PCR. In medicine, it enabled the diagnosis of genetic disorders, the detection of infectious diseases, and monitoring of gene therapy. In agriculture, PCR aided in the early detection of plant diseases and the creation of genetically modified crops. And in conservation, it helped in the identification of endangered species and the study of biodiversity. Mullis himself received the Nobel Prize in Chemistry in 1993 for his invention, and since then, PCR has become one of the most widely used techniques in molecular biology. PCR allows scientists to take tiny amounts of DNA and amplify them into millions or even billions of copies. This breakthrough had a profound impact across various fields. This infusion of capital created an entire industry focused on manipulating life's building blocks. In 1974, Rudolf Janisch inserted a gene from the mouse into a bacterium. In 1978, he shocked the world by modifying a mouse embryo and creating the first genetically modified mammal. In 1980, Cohen and his team used a modified bacterium to create the first genetically engineered plant resistant to a common herbicide. All these advances happened within just a few years, fueled by the realization that DNA manipulation wasn't just possible, it was practical, opening the floodgates to biotech research. Gel electrophoresis separated DNA fragments by size, like a molecular sieve. Restriction enzymes sliced DNA at precise spots, acting as molecular scissors. By the 1980s, at least 300 biotech firms existed globally, many focused on developing genetically modified organisms for research or commercial use. And by the late 1980s, the biotech industry was valued at over $20 billion. Gentech alone was worth over $10 billion. This rapid growth brought immense wealth to investors, founders, and research. Suddenly, DNA was not just a scientific curiosity. It was a commodity, a potential gold mine. Cell cultures allowed scientists to grow and study cells outside the body, and by the end of the decade, scientists had fully cracked the genetic code. They could now read DNA instructions, translating the sequence of nucleotides into amino acids. But reading DNA was only the beginning. Scientists started wondering if they could also rewrite it. Was it possible to edit the very source code of life? The image of the helix had inspired art and identity, but its power lay deeper still. DNA was no longer just a message. Paul Beck at Stanford, and, independently, Herbert Boyer and Stanley Cohen at UCSF developed techniques to cut and splice DNA from different organisms. By inserting a recombinant DNA strand into bacteria, scientists turned them into biological factories. And what did they make? Some might have guessed proteins, the workhorses of the cell. But in 1978, something far more valuable hit the market: human insulin. Until then, diabetics relied on insulin extracted from pigs or cows, which triggered allergic reactions. Now, thanks to recombinant DNA, Eli Lilly and Company produced the first synthetic human insulin in 1978, replacing animal-sourced versions. Soon after, they produced other essential proteins like human growth hormone and tissue plasminogen activator to dissolve blood clots. Using restriction enzymes, molecular scissors that could cut DNA at specific sequences, they could excise certain genes. Then using ligases molecular glue, they could reassemble these pieces end to end, even if they came from totally different organisms. This merging of DNA from multiple sources created recombinant DNA, the raw material of genetic engineering. It was something we could edit, replicate, and own. This chapter traces the rise of biotechnology and how the double helix became both blueprint and battleground. The answer came in 1972 when two scientists changed the game. But where did this new DNA go? Enter bacteria, nature's tiny gene carriers. At the same time, other labs made significant strides, but it was also big business. In 1976, Genentech was born, the first biotech company. By the late 1970s, several companies were producing human proteins, like insulin and interferon, using recombinant DNA technology. Within years, Genentech produced synthetic human insulin, replacing animal-sourced versions, developed clotting factors for hemophilia treatment. For instance, in 1980, the US Supreme Court ruled that genetically modified organisms could be patented. PCR, the copy machine of biology. In 1983, biotechnology got a turbo boost. In forensics, a single drop of blood or a few strands of hair could now provide enough DNA for analysis, revolutionizing criminal investigations. And as PCR became more accessible and cheaper, its applications grew exponentially. But the story of DNA doesn't end there. Before we leave, here's your summary and key takeaways. Now, here's your quiz. Test your knowledge of the key concepts covered in this chapter. DNA is everywhere these days. There are DNA tests to tell you which Hogwarts house you belong to. You can order custom t-shirts with your own genetic code printed on them. Even your washing machine might use enzymes that originated from bacterial DNA. But when the character of J.R. Ewing was shot in the iconic American TV series Dallas, viewers across the nation were glued to their screens, waiting for the reveal of his assailant. And when the perpetrator was revealed to be the seemingly sweet and innocent Kristen Shepherd, fans were shocked. Like fingerprints or ballistics, DNA evidence can be used to place individuals at crime scenes. In fact, it can even exonerate those who have already been convicted. That's why, since 1986, over 375 people have had their convictions overturned because of DNA evidence, most of them in the United States. With a simple test, we can find out where our ancestors came from, what they looked like, and even what diseases they may have suffered from. In fact, it's one of the most common fears among people who have had their DNA tested. But what happens when the spotlight shines on our own DNA? But what if the story of Noah's Ark was not just a religious tale, but a reflection of real events? That's why we often don't know how or why certain traits evolved over time. But DNA can help fill in the gaps in our knowledge. As we learn more about our genetic makeup, we'll be able to develop new treatments for diseases, trace our ancestry, and even customize our own DNA. But with these advances come new ethical challenges. That is why it is so important that we continue to have open and honest conversations about the implications of DNA technology. The more we understand about DNA, the more we can harness its power for good. So let's keep exploring, keep learning, and keep pushing the boundaries of what's possible. Who knows what we might discover next? By comparing the DNA of different populations, scientists can identify genes that have changed over time and learn more about the evolution of our species. We've covered a lot of ground, from the double helix to the latest research and everything in between. If you enjoyed this chapter and want to keep learning about the incredible science that surrounds us, then don't forget to subscribe and join me on my next adventure. Until then, keep asking questions, keep exploring, and keep thinking like a scientist. For example, a study published in 2018 used DNA analysis to track the evolution of lactose tolerance, the ability to digest milk sugar. The researchers found that the ability to digest milk sugar evolved independently multiple times in human history, and it spread rapidly in populations that kept livestock. This suggests that the ability to digest milk sugar was beneficial to our ancestors, giving them a source of nutrition that few other animals could take advantage of. Another study published in 2021 used DNA analysis to track the evolution of skin color in humans. The researchers found that darker skin tones evolved independently in multiple human populations. After they migrated out of Africa and into regions with less sunlight, this suggests that darker skin tones provided an advantage in these environments, protecting our ancestors from the harmful effects of the sun. Overall, these studies show that DNA can help us better understand the evolution of our species and the traits that make us who we are. Scientists have long believed that humans experienced a genetic bottleneck after the last ice age, when temperatures rose rapidly and glaciers melted, causing widespread extinctions. But until recently, there was little evidence to support this theory. However, a study published in 2015 used DNA analysis to shed new light on the issue. Researchers studied the genomes of more than 1,000 people from around the world and compared them to the genomes of ancient humans from 10,000 years ago. They found that there had been a significant reduction in genetic diversity during the last ice age, suggesting that the population had indeed experienced a bottleneck. Furthermore, the team discovered that the majority of genetic variation found in modern humans today actually arose after the Ice Age ended, which means that the period following the Ice Age may have been a time of rapid diversification for humans. This finding suggests that our species may have experienced a population explosion after the Ice Age, leading to the emergence of new genetic variants and traits. This could explain why humans were able to expand into new territories and develop new technologies after the Ice Age, setting the stage for the rise of civilizations. Overall, the study provides a fascinating glimpse into our species' past and helps us better understand the forces that have shaped human history. As DNA testing becomes more commonplace, it's important to remember that we don't want to lose sight of our own privacy. That means being careful about who we share our DNA with and making sure that our genetic information is protected. After all, our DNA is one of the most personal things we have. It's literally what makes us who we are. So let's make sure we take the time to protect it. But here's the thing. DNA isn't just unique to you, it's also unique to your family members. That means that if someone steals your DNA, they could also end up learning information about your relatives, which raises some really interesting ethical questions. For example, what if someone stole your DNA to find out if you had any genetic relatives who were also donors? Or what if they found out that you had a close relative who was a suspect in a crime? These are all possibilities that are becoming more and more likely as DNA testing becomes more popular, and it's important to think about them as we move forward. But DNA can also be used to connect us to people we never knew existed, like the case of Sandra London, a woman who discovered she had a half-sister she never knew about, or the case of Brian John, a man who discovered he wasn't related to his father or his half-brother by blood. These kinds of stories are becoming more common as DNA testing becomes more popular. And while it can be a shock to learn that your family tree looks different than you thought it did, it can also be a wonderful thing. Just ask Julie Little, a woman who discovered she had a half-sister she never knew about, or Brian John, a man who discovered he wasn't related to his father or his half-brother by blood. These kinds of stories are becoming more common as DNA testing becomes more popular. And while it can be a shock to learn that your family tree looks different than you thought it did, it can also be a wonderful thing. But forensic DNA isn't just about catching bad guys. It's also revolutionized the way we deal with missing persons and unidentified remains. In the aftermath of disasters like 9/11 or the Boston Marathon bombing, DNA analysis is used to identify victims when other methods fail. And in cases where traditional identification methods aren't enough, like mass disasters or cases involving unidentified remains, DNA can provide the crucial link needed to bring closure to families. After all, she seemed so nice, but upon closer inspection, maybe not so much. Today, the industry is worth billions of dollars and has given millions of people around the world an opportunity to learn more about their family history. But while some people are happy to simply know their ethnic composition, others use these kits to find living relatives or uncover surprising secrets about their biological parents. In fact, these kits have even been used to solve cold cases by connecting law enforcement with distant relatives of suspects. In 2018, for example, a man known only as the Golden State Killer was arrested after a relative's DNA was uploaded to a public database and matched with evidence from a crime scene. It was not long before Kristin's true colors shone through, and she went back to her old tricks. Imagine finding out that you have a half-sibling you never knew about, or that your parents were not actually related by blood. For many people, these revelations can upend their sense of identity and change their view of themselves and their relationships forever. But despite these challenges, the desire to know one's roots runs deep, and genetic genealogy has given millions of people a chance to uncover the secrets of their past. So it should come as no surprise that DNA features heavily in our stories, both those we tell ourselves and those that are told about us. But it turns out there might actually be some truth to fiction after all. In a study published in 2015, researchers at the University of Texas at Austin found that people with a genetic variant linked to low levels of serotonin were more likely to be aggressive and reoffend than those without it. And guess who had this gene variant? Kristen Shepherd herself, aka actress Dana Wynn. While the study didn't directly investigate the actor's behavior, it does provide an interesting perspective on her character and the genetic basis of criminality more generally. But how did this molecule go from being confined to dusty textbooks to becoming so deeply ingrained in our culture? In this chapter, we will see how DNA has gone from being a scientific curiosity to becoming part of our everyday lives. We will also learn how this molecule has been used to catch criminals, find long-lost relatives, uncover our personal histories, and even help us find ourselves. Let's get started. Who shot JR? Okay, maybe that question is a bit dated. If this story sounds familiar, it's because the plotline of Good Girl Gone Bad is one of the oldest in Hollywood history. This brings us to our first example of how DNA has made its way into everyday life. Forensic DNA. Family secrets. A new generation, the birth of genetic genealogy began in 2000. when two men named Gedmatch and 23 launched DNA testing services to help people find their roots. But while these discoveries can be life-changing, they can also be overwhelming. Lost and found, one of the most amazing things about DNA is its ability to connect us to our past. Identity theft. Do you ever worry that someone might steal your identity? Well, if you do, you're not alone. Privacy Watch. In today's digital age, it seems like we're constantly bombarded with information about celebrities and public figures, whether we want it or not. After the fall, according to legend, the biblical figure Noah survived a great flood by building an enormous boat and taking his family and 2 of every animal species on board. Evolution in action. Humans have been around for hundreds of thousands of years. But it wasn't until relatively recently that we started to keep records of our history. A world of possibilities. The future of DNA is full of possibilities. Hey, thanks for joining me on this journey through the amazing world of DNA. Chapter 14. The Double Helix and the Road Ahead. A future written in code. In the beginning was the helix. Now we're writing the sequel. It began as a riddle of heredity, a mysterious molecule, coiled like a whisper, hidden in every cell. Watson, Crick, Franklin, and others gave it shape, and in doing so, they gave the world a new mirror. In 2010, scientists at the J. Craig Venter Institute created the first synthetic genome. They wrote their own 5,000-letter sequence onto a bacterial cell and transferred it into a new life form. A new kind of organism was born. The double helix-crossed, spiraling. Heredity is a symbol of connection. It links indigenous roots to global migrations. It bridges illness to understanding. In 1953, 2 young men stared at a model of sticks and wires and glimpsed something timeless. In 2025 and beyond, we stare at glowing screens and digital readouts. Yet the wonder is the same. DNA is no longer just the molecule of life. This episode and the entire 14-chapter season were made possible by our generous supporters. From the earliest civilizations to the modern era, we've pondered our place in the cosmos. They revealed that this helical staircase held the very essence of life, the genetic code that determines everything from our eye color to our susceptibility to certain diseases. This discovery revolutionized our understanding of biology and ushered in a new era of scientific exploration. And with each passing year, we delve deeper into the intricate code that makes us us. We have explored the vastness of our genome, the entire instruction manual of human life. We have uncovered the secrets of inheritance, explored the depths of our genome, and even ventured into the realm of synthetic life. But this story is far from over. There's still so much more to discover, so many more frontiers to explore. Who knows what wonders await us in the uncharted territories of DNA? Perhaps we will unlock the secrets of aging, unravel the complexities of the human brain, or even engineer new forms of life. The possibilities are truly endless. We have encoded messages into DNA strands, essentially leaving invisible marks on the building blocks of life itself. Together, we can uncover the mysteries that lie within our genes and harness the power of this remarkable molecule to improve our lives and the lives of future generations. Let's embark on this thrilling journey and see where the future of genetic discovery takes us next. This might sound like science fiction, but it's reality, and it raises profound questions about the nature of life and the limits of scientific exploration. We have identified genes linked to diseases, shedding light on the origins of ailments that once shrouded in mystery. And we have harnessed the power of genetics to tailor treatments to individual patients, ushering in an age of personalized medicine. It all started with a simple question. What makes us who we are? Was it the environment that nurtured us? The society that shaped us? Or was there something more? Some hidden blueprint that dictated our traits and characteristics. Enter DNA, the molecule of life. It's a microscopic marvel, a double helix composed of 4 chemical bases that holds the key to our existence. To learn how you can support this production and all the creative work we do here, visit our support page. That's how you make a lasting impact. You leave a legacy that supports creativity, inspires minds, and builds a better tomorrow. Because, just like DNA, the future of this channel is written in code. It's code woven into the fabric of our existence, shaping who we are and how we interact with the world around us. Gene editing technologies like CRISPR have given us the power to alter DNA sequences with precision. This capability has opened up new frontiers in medicine, allowing us to correct genetic defects and potentially eradicate inherited diseases. But it also presents us with ethical quandaries. As we wield the power to modify the building blocks of life, we must tread carefully, ensuring that we don't alter the delicate balance of our existence. It's the map of what we are, the tool to heal, the spark to dream, and like the helix itself, it twists endlessly, always upward. Thank you for taking this journey with me across 14 chapters. This ends the story of finding DNA in human history. It binds us, thread by thread, to one another and to the Earth itself. I have a favor to ask you. Please write a review of this podcast. You can do it right now by clicking here, or if you prefer, you can find the link in the description below. It only takes a few seconds and really helps other people discover this podcast. Plus, we love hearing what you have to say, so please tell us what you think. What did you like? What would you change? What surprised you? What do you want to hear more of? By sharing your feedback, you will help make this podcast better. And if you haven't already, be sure to subscribe because more adventures in human history are just one click away. It reminds us that life is not a monologue. It's a duet of inheritance and environment, code and choice, past and possible. We can now encode books, photos, even movies, in molecules smaller than a grain of sand. Researchers are developing biocircuits, genetic switches that act like logic gates, mimicking computers within living cells. Nature's code has become a programmable platform. Today, DNA is more than biology. It's infrastructure, identity, and frontier. Imagine an altered mosquito population that eradicates malaria or stops a beetle from carrying chestnut blight. But gene drives could also disrupt fragile ecosystems. If every child born in the next century has a genetic risk score, who will have access to reproductive technologies? How will predictive genomics affect insurance, employment, or even law? As we peer closer and closer into the building blocks of life, we must ask again, should we? Who decides? Can we undo what we edit? The future calls for ethics as agile as innovation. The helix hasn't stopped twisting. It's spiraling into the future. The ongoing revolution. Science didn't pause at discovery. The Human Genome Project, launched in 1990 and completed in 2003, mapped the entire 3 billion letter instruction book of Homo sapiens. Genome-wide association studies, or BRs, now link traits and diseases to specific genetic regions. Personalized medicine uses your own DNA to tailor treatments from cancer therapy to mental health care. We are decoding ourselves faster, cheaper, and deeper every year. Synthetic, life, and DNA computing. DNA is now material, not just message. The next moral maze. With great knowledge comes great peril. Gene drives can alter whole species. Unity in the code. For all our differences, we are 99.9% genetically identical. Epilogue. A helix without end. But please don't consider this the end of our story together. The incredible science behind DNA took decades to uncover, But today's technology lets us explore it like never before. For millennia, humans have been deciphering the language of genes, unlocking the secrets of inheritance and the mysteries of life itself. But unraveling its secrets wasn't easy. Like an ancient scroll, DNA remained hidden for centuries, until the brilliant minds of Watson and Crick finally deciphered its structure in 1953. But the story doesn't end there. We've come a long way since then. But it gets even more fascinating. We've ventured into the realm of synthetic life, creating artificial DNA and rewriting the very code of life. We've delved into the ethical dilemmas posed by these advancements. Our journey through the world of DNA has been nothing short of extraordinary. So join us on this incredible adventure as we continue to explore the fascinating world of DNA.